Okay, good morning and welcome back to the second session of the eucleg workshop. In this session we'll have three talks given by three of the species leaders. We have a talk on soybean, a talk on pea, and finally a talk on faba bean. And I'd like to introduce Hilde Muylle for the first talk. She'll be talking on genomic assisted breeding in soybean. Okay, thank you, Hilde.
Okay, good morning, everybody. I have the honor to give the talk on soybean, but I have to say that this is in fact the work of Jonas Aper, who was a crop leader within eucleg on soybean before he moved to a new position. So genomic assisted breeding in soybean, maybe first an introduction on the crop itself. Soybean, indeed a very important crop, as it has a high protein content, up to 40% in the seeds and 20% of oil. So a very important crop for food and feed applications. And it originated indeed from East Asia. My presentation is back. So it has been domesticated already for a long time, 7,000 to 9,000 years ago. The domestication started. And if you look to the worldwide production area and the worldwide production volume, this is still increasing. So there's a high demand on soybean. And if you then look to the proportion of soybean that has been produced or is produced in Europe, this is only a very small proportion. It's only 2.3% of the volume that is produced within Europe. However, we consume quite a lot of, and we need quite a lot of soybean within Europe. So currently we produce only 5.5 million hectares of soybean in Europe. And this meets only 1/3 of our need for soybean. With this increasing demand for protein and the demand to get Europe protein self-sufficient, we need, there's an interest to grow soybean or to increase the production of soybean in Europe. When we look where those 5.5 million hectares are situated within Europe, you can see it's mainly in the belt at a latitude of 45 degrees. So indeed, in there's the region where current varieties can grow and where they can grow profitable. But if we want to increase the production area within Europe, we should aim for varieties which are suitable to produce within the European community. And so when we want to increase the acreage, we have to move more up north and to look for varieties that can grow in those conditions, which are mainly characterized by a colder climate, but also a shorter growing season. So indeed, we have to adapt those varieties to those conditions. Which means that, first of all, we have to improve yield and to make varieties that really can profit from this short growing season. And how can we achieve varieties that can grow in those shorter regions in the Nordic areas is to select for varieties where the cardinal temperatures are more adapted to those regions. So we should have varieties that can germinate at colder temperatures. Also, a second characteristic where we should breed for is a photoperiodicity. So soybean is a short day crop, so it should sense short days before it turns to generative status. And it's known that there are some varieties that already can shift to generative status in conditions in higher latitudes. But if you want to go further up, we need even photoperiodic insensitive varieties. So indeed, selecting varieties which are less sensitive to photoperiodicity, these are ones are needed. And a third item which we should improve on for Northwestern Europe adapted varieties is on determinacy. Because we have those cold conditions or cold spells during the season, we should aim for plants which are not determinate. So if there are cold spells and flowers are getting destroyed, we can still expect a second flush of flowers and to have a production after the cold spell. So these are three major breeding goals within the European soybean breeding programs. Further, we need as well to have adaptation towards biotic and abiotic stress tolerance, drought tolerance indeed. We are suffering from the last years from drought spells, so drought periods. As I told already, cold tolerance is very important characteristics where we should breed for, and also disease resistance. So we know for some breeding lines that there is a susceptibility to Pseudomonas, and we should prevent that this comes into the germplasm of Northwestern European soybean. So having those breeding goals, how can we achieve or how can we achieve those new varieties, those breeding goals? This is expressed or summarized in this formula. So how to get genetic progress? In fact, we should have traits where we can select for. So traits that show high heritability. We should be able to imply a selection index. So the I stands for the selection intensity. And we can only increase the intensity when we have a large pool where we can do the selection inside, together with a lot of phenotypic phenotypic diversity. So the I and the sigma is really on the variation which is needed to select on. And then to achieve a quick genetic gain, we should aim for a reduced breeding cycle time. If you look to the current breeding program of soybean, in fact, it's a self-pollinating species. You take two homozygous lines which are crossed, then you go to an F1, and through single seed descent, you go up to F4, and then from then on, you can start to do phenotyping at plot or row level, so where you can assess the new genetic combinations in at row level. And after further selfing and further upscaling of the seed, you get an F9 a line which can be released as a variety. So it takes up to 9 to 10 years before a variety is produced. So if you could, if you go back to the previous slide, we could impact the breeding cycle time, we could achieve a bigger or a quicker genetic gain. And that's what we aimed for in eucleg, to see whether we can use genomic selection to speed up that breeding cycle. So here again, you have the breeding scheme of soybean. And if we could, instead of phenotyping from F4 on, if we could do there a selection on the basis of genotyping instead of phenotyping, we could speed up the breeding cycle about 3 to 4 years. And that's what we investigated, or within eucleg, whether we could set up genomic prediction equations to predict the potential of a given plant on their phenotype. Yes. The way we did this is, first of all, by collecting the correct gene pool which is necessary for breeding. So as I told already in my introduction on soybean, we have to use early maturing or very early maturing material to be able to produce seeds in time in the short season we get in Europe. So and worldwide, we categorize varieties in maturity classes. There are about 14 defined. And in fact, in Europe, only the very early ones, the 4 classes which are set here on the slide, can be used for growing in Europe. So in fact, it's within those, in that gene pool that we selected the material for eucleg. Also, as I told already, to prevent the risk of a total loss of the yield, we aim for semi-determinate growth, so where we can have consecutive flushes of flower, and it should be a material which is cold tolerant to grow in this Northwestern European conditions. We already knew from beforehand that the current European varieties, they are based on a very narrow genetics. They have been selected from already pre-selected material, and we knew already on beforehand that we should enlarge our collection with additional material. So that narrow genetic base is indeed caused by the number of ancestors that were used to build those varieties. And we should take care of potential selective sweeps that have occurred during the previous selection cycles. The collection of eucleg, it consisted of 480 accessions, as I told already, from different origins. It's represented here. We paid attention to have breeding line varieties, but also land races and more older material. It was also derived from different origins, also different breeding programs donated material within this collection. And so finally, we ended up with about within a collection of 480 accessions. The first thing we did is to assess whether this collection was harboring enough genetic diversity for the work we intended to do. And we compared the collection of eucleg with already described collection from China. So we compared the genetic diversity of eucleg with the collection of 394 accessions that contained about 120 wild accessions of soybean and 270 cultivated varieties. The way we analyzed this genetic diversity, we used for that a 355K soybean SNP chip, and we investigated the genetic diversity on those 805 genotypes. This SNP chip was highly useful within eucleg. In fact, it yielded 285,000 SNPs available for genetic diversity studies. We ended up with about 23 SNPs on a 100 kb region on average. We observed that ALD decay was about 55 to 188 kilobases, and we had a very nice nucleotide diversity between 0.23 and 0.31. Looking at the population structure and using those markers, we were able to identify the three different collections. So the wild accessions clearly differentiated from the cultivated Chinese material, and eucleg also clearly differentiated from the Chinese cultivated material. So indeed, we had a quite unique material, which we could differentiate from the wild. So it's already some evolved material we were dealing with. And looking within our eucleg collection, we could distinguish about 5 groups, and they were mainly related towards the breeding programs they were coming from, or the region they were selected in. So the first one was mostly South European material, which was later than for example, the group 5, which was very early, which is indeed very early material. The fourth group is mainly consisting of edamame types, indeed. So this is the vegetable soybean, which is eaten fresh. And this material was indeed distinct within the eucleg collection. Then the groups 2 and 3. So 2 is really originating from Eastern Europe, and the third group from Western Europe, containing early to very early material. So from this marker study and genetic diversity study, we used this as well to look whether we have selective sweeps. So we looked to the diversity in our collection compared to the Chinese wild material. And we identified 23 selective sweeps in our collection, in fact affecting 4% of the total genome length, which we should take care of. For some of the selective sweeps, we already knew on beforehand that they might have been there in our collection. For example, on the maturity genes, we knew that we had very early maturity material in our collection, and that those genes coding for early maturity might have been fixed in our collection. Also, other selective sweeps were identified, and they were mainly located where also QTLs for important agronomic traits that have been selected for already are situated. Yeah, so that was a conclusion from our genetic diversity study on the eucleg collection. So indeed, there is a reduction in genetic diversity in our eucleg collection compared to the wild collection. We observed also a longer LD than in the wild material. We observed some selective sweeps. However, determinacy, we expected it also to be fixed in our material, but it wasn't. So we couldn't observe any selective sweep on that region where determinacy is coded. So for us, although 4% of the genome is affected by selection, we assume that there's still enough diversity for breeding within this collection. So next, having this gene pool within eucleg, it was phenotyped quite extensively. So it was phenotyped in the multi-location field trials, and as already explained before, in augmented design at 4 different locations, with some sub-collections higher up in north in Germany and more south to Europe. And the 2 main sites were more situated at latitude 50 degrees. So multi-location field trials next to controlled conditions. So the collection was also phenotyped for cold tolerance during germination, drought tolerance, and disease tolerance. What we observed in our collection, that indeed the later maturing material, so to the left you can see the maturity in relation to seed yield. You can see that the later the material is, the higher the yield. So indeed, the early maturing material is yielding less. So there we need for sure yield improvement to have a profitable crop. But what we observed as well, we could not move forward to later maturing material because those genotypes don't reach full maturity or full potential in our conditions. So we have to move in between the early and so to take into consideration the maturity of the material to obtain sufficient yield. Another observation we made in our collection is that we have quite low yield stability. So they were phenotyped during 2 years. And if we look to the correlation between 2018 2019, we see that there's a very low correlation between both years, indicating that yield stability is also a problem within our material and that we should aim for or should breed for yield stability as well in our conditions. So we observed phenotypic difference, phenotypic diversity and yield. Also for drought tolerance was assessed. We looked to the reaction to drought during the generative phase. So we looked to canopy wilting, leaf senescence according to drought or as a reaction to drought. In there, we could see that indeed 2018 was a different season than 2019, but we could observe phenotypic diversity in canopy wilting and also in leaf senescence, showing a nice heritability, especially in the later observations after drought. But we didn't see a lot of diversity in canopy temperature. So in the crop water stress index, we didn't see a lot of genetic or phenotypic diversity. Another abiotic stress that was assessed during eucleg was the tolerance to cold inhibition. So when we want to grow soybean at higher latitudes, we want that the germination goes smoothly, even under cold conditions. And this was assessed under controlled condition. In there, we could use high throughput phenotyping platforms, because this is also a message I want to address. Within eucleg, some high throughput phenotyping platforms were established, allowing to assess a higher number of plants and allowing to assess a higher phenotypic diversity. This is also one of the factors in the famous formula of genetic gain. By means of those phenotyping platforms, we can assess a higher number of individuals. If we look to the tolerance to cold inhibition, indeed, we could see phenotypic diversity in the reaction towards cold, so during germination, and also the formation of normal seedlings was impacted. And we could see that there was a link with the maturity groups. Indeed, the ones which were late maturing were more affected by cold during inhibition, during germination. However, we saw phenotypic diversity, so we could go for breeding in there. Another high throughput phenotyping platform I would like to illustrate. So during eucleg as well, we used UAV methods to assess phenological parameters of soybean. So here you can see the different pictures taken during the growth of a control experiment at Ilvo. So you can see that the germination and canopy closure to the upper series of photos of pictures, and at lower you can see then they move towards senescence, and you can really see the diversity already by eye. But by means of flying different times over those control fields, we can simulate the growth curve of each genotype. And from that growth curve, we can derive different phenological parameters interesting to select for. Because yield is a very complex trait, but by means of UAV methodology, we can dissect yield in different yield components and different yield aspects. And from there, we can assess, for example, youth growth, so the growth canopy closure, time to close a canopy, the time to needed to senesce, final plant length, and so on. So this also provided a mean to assess quite a lot of phenotypic traits in which we observed phenotypic diversity. So assessing the collection revealed that there was quite a lot of phenotypic diversity and in which we could select for. But we wanted to know as well the genetic control of those traits. What genes are related to phenology? What genes are related to architecture, tolerance to biotic and abiotic stress? And for sure as well, what is the covariation with other traits? Because what we have seen already, for example, if you would select for tolerance to cold inhibition, you might select for the early ones, but preventing the selection of the late maturing ones. So there's some covariation with important traits, and we should know the genetic control of each trait and the interrelationship with other phenotypic traits. The GWAS studies which were performed within eucleg, so this was on the soybean collection I have been describing, using the 355K soybean SNP chip, and we looked whether we found or we checked the association with given genomic regions for each trait. Let me go through some of the results. So indeed, for flowering and maturity, although we had some selective sweeps on the E2 and E4 gene, we could identify some other genes related to flowering and maturity. So to the left, this is on flowering. We identified clearly an association with E1. And for maturity, we had some interesting candidate genes, which we can study more in depth and to see what the role is on maturity within the collection. So for flowering and maturity, we had some interesting candidates. Similarly to tolerance to cold inhibition, we see some regions popping up and explain quite a lot of phenotypic variation for those markers. So further research is going on to identify what genes are behind those significant associations. And they might set light to the biological processes behind it. Similar for disease resistance, here we see some two regions popping up on Pseudomonas resistance. And finally, on drought tolerance, we identified also some significant associations. And behind some of those significant associations, we know already, we have some reported QTLs already on water use efficiency, for example. So indeed, we have some quite interesting associations, significant associations with important traits. But how can we implement this now in the soybean breeding program? One way is to use this knowledge in our genomic prediction procedures. So, in fact, that's what we tried as well within eucleg to build prediction formulas to predict yield and protein. Oops, sorry. So to predict yield and protein. So we had our collection, which we divided in a training population, which was phenotyped and genotyped. We made a prediction model, and then we tested whether this prediction model was able to be applied on the test population and to see whether how good the prediction can be solely on the basis of genotyping. Some results from the soybean collection. So we tested how big the test population should, the training population should be. If we divide the eucleg gene pool and we take only 25% as test population and 75 as training population, we get a good prediction for protein and seed yield. But you can see the further we go, so if we have 75% of our population as a test population and only 25 as training, we see that the predictability is lower, although it's quite high in the case for a protein and seed yield. Quite promising results here to see that we have quite good predictabilities. Next, as we know that in the eucleg collection we had some structurations, so there were quite clear distinct groups. We tested whether the prediction or the prediction equation was built within a group and used to predict the phenotype for the rest of the population. And you can see that depending on which group was used to build the prediction equations, that predictability is changing. So indeed, some of the groups, they're better suited to build a model, and they're better they're providing a more robust prediction equation to predict the phenotype. So very important is to know the structuration within the population and to see how this can be used to make your predictions. And the last test that was the last but one test that was performed within eucleg was to look to the marker density. So we had about 285,000 polymorphic SNPs, but in fact we could reduce it up to 2,000 SNPs to have still quite good predictability. So the size of the training set was much more important to have a good predictability compared to the numbers of SNPs taken to build the prediction equation. So it could be reduced even to 2,000 SNPs and having a quite similar predictability. And the last thing, as we had the GWAS methodology within eucleg, we tried to put GWAS, so to put associated markers with protein or with seed yield as fixed factors within our prediction model. And what we could see, if we put already known associated markers or already known genomic regions as fixed factors within our prediction model, we can achieve higher predictabilities compared to whether to a completely random driven model. So coming to a conclusion on the genomic prediction models, indeed within eucleg, the size of the training set is quite important, as well as the structuration within the population to take this into account. Very interesting to see was that the marker number needed can be quite low, and that associated markers already known to be associated with the trait, if you put them as a fixed effect, this aids the predictability. And of course, we see a difference between traits. Some traits are have a higher heritability than others, and this is reflected in the prediction efficiency. So indeed, how can we put this into the breeding program? In fact, if we have those prediction equations available, we can test already at F4 stage the genotype of each descendant, so of the within the breeding program, to genotype it and to predict the phenotypes. And in fact, at that time, we can already have an idea of the phenotypic performance of that genotype, and we can already select new parental lines that feed into the parental crosses. So in fact, instead of waiting up till F8, F9, or so by the end of the selection cycle, we can shorten the cycle by 3 to 4 years by selecting already at F4. So concerning genomic selection, then how to put it into the breeding program. So it's important to have the correct and then the suitable training set within your breeding program. So there are different ways to set up the breeding program. You can select within populations. So if you make pair crosses, you continue to select within those ones, or you can select between them. And so simulations already published by other authors. So within population selection are assumed to be superior to across family selection, which is quite logical, I think, because then you really select within your training population. Also concerning the renewal of calibration curves, so it takes some time to build up those calibration or prediction equations, and within simulations, it has been shown that the real potential of a prediction curve is reaching its maximum or the highest gain after 10 cycles of breeding, and after that, the training set should be updated. Of course, it can be updated during the consecutive breeding cycles, but after 10 cycles, for sure, the maximum is out of it. And a third important issue is when you select within the population, you have to take care that you continue to maintain genetic diversity within your collection. Because indeed, after genomic selection, you go in a certain direction, and you should take care of not losing too much diversity within the breeding program. Looking also to soybean and going back to the start of eucleg and where we are now, I think a lot of new tools become available. A lot of knowledge is becoming available within soybean, mainly in outside Europe, but I think also within Europe, quite a lot of knowledge has been generated. But one thing is on the knowledge which is available now on soybean genomes. Quite a lot of whole genome sequencing has been done, shallow whole genome sequencing, but there's quite a lot of data available which can be used to do haplotype discovery within those genotypes. Also, next target candidate genes, quite a lot of genes involved in those important traits we are selecting for. They have been characterized. We know the different haplotypes available in those candidate genes. So this knowledge can also be integrated within the breeding programs. So and then next also the training set, how to build this and how to renew it. This is quite important for especially for Northwestern Europe, because we don't want to be too narrow within our collection. So I think in the 4 or 5 years of eucleg, during that duration, quite a lot of new technologies and knowledge is becoming available, having good perspectives for genomic selection within soybean. And that's what I wanted to end with. Thank you.
Thank you very much, Hilde, for that excellent presentation. Very comprehensive. We have a number of questions. The first question is, would it be more efficient to apply genomic selection from the F3 stage rather than the F4?
Good question, Stephen. I think in there we still have quite a lot of heterozygosity, so that's something we could test in simulation. Would be happy to discuss on that with you.
And another question was, you talked about reducing the number of SNPs and how were they selected at random when you reduced the number of SNPs that were used, or were they selected based on their position on the genome?
Yeah, so they were based on the position on the genome. They were nicely dispersed within the genome, also based on the local LD within that region.
Yeah. Okay. And some two more questions. So a question about the diseases that we have in Europe and whether how they compare worldwide. So are soybean mosaic virus and also insect pests serious problems for soybeans in Europe?
Yes, indeed. So the Soybean mosaic virus is indeed a problem within Europe for sure. Yeah, nothing more to say then.
And are there insect pests as well? Or?
Not to my, I'm not that familiar with the diseases in soybean, but to my opinion, no. If some breeders are in here, they can reply on this question themselves, but I'm not a specialist on that.
No problem. And then one final question is, which was about you early on in the talk, you talked about indeterminacy. So would selection for indeterminacy lead to problems with plants never ending flowering and then having a low harvest index?
Yeah, indeed. In fact, we don't select for indeterminacy, but semi-indeterminacy. So there's a gradient between determinate and indeterminate. So we try to select for semi-determinacy, where we can allow a few fluxes of flowering. That's it, not that much. Because if you go for indeterminacy, indeed, plants don't senesce. They continue to make new flowers and new nodes. No. So maybe the last question I see as well on the subset. The subset was again 100. Yeah, thank you, Bernadette. 100 accessions more north and 100 accession south, but they were not the same. So these were the 100 adapted to the north and 100 more adapted to the south. Okay.
Okay. Thank you very much.
You're welcome.
I know we have a new question. How is the genetic control of determinacy in soybean?
How is the genetic control of determinacy? There's one major locus involved. It's the DT gene. And then we expected this one to be fixed within our material, but wasn't. Yeah, let's answer the question.
Okay, thank you very much. It is time to move on. We'll just have a couple of moments whilst before we move on to the next presentation.